Romain GUIÈZE.
Hi.
Hello, Romain.
Hello.
So Romain is working in Clermont-Ferrand University at the hospital in the Department of Clinical Hematology and Cellular Therapy, and is focusing on leukemia and lymphocytic leukemia and therapeutic resistance. And he tried to find out how to best benefit from novel agents by using genomic monitoring. So thank you to be here, Romain, with us, and take the stage, even if it's in virtual.
Okay, thank you very much. Hi, everybody, and ni hao is the only word I can say in Chinese. So thank you very much for inviting me to talk about my work. So as been said, I'm a hematologist, a physician working in the Department of Hematology in Clermont-Ferrand. And I'm also involved in research by leading projects related to clonal heterogeneity and therapeutic resistance in chronic leukemias. So here today I'm going to focus on this work, mitochondrial reprogramming as underlying resistance to BCL2 inhibition in lymphoid malignancies. So as you know, mitochondrial apoptosis is governed by the BCL2, B-cell lymphoma 2 families, including both pro and anti-apoptotic protein. And across cancer, apoptic deregulation can result from overexpression of the apoptotic BCL2 protein that can sequester certain pro-apoptotic BH3-only proteins, such as BEAM and BEAD, to avoid oligomerization of pore-forming proteins such as BAX and BAC and subsequent mitochondrial outer membrane permeabilization. And through cytogenetic alteration, BCL2 is overexpressed, and there is also an important BCL2 dependency in lymphoid malignancies, and BCL2 has been a rational therapeutic target for such reason. So now we have this compound, the venetoclax, that is the first-in-class BCL2 inhibitor, and that has been FDA approved for the treatment of chronic lymphocytic leukemia. And this compound acts actually by displacing the pro-apoptotic BH3-only proteins such as BEAM from BCL2 and allowing them to bind and activate proteins such as BAX and induce the cytochrome C release and the final steps of mitochondrial apoptosis. So venetoclax has demonstrated potent clinical activity, but disease progression and therapy occurs in around 30% of relapsing patients at 15 months and is expected to become a major challenge in the next future. So thus a critical question in the field is how we can identify mechanisms of BCL2 inhibition resistance in lymphoid malignancies. So you can find more of this data in the related publication in Cancer Cells a few months ago. So we address this through three complementary approaches. First, we analyze whole exome data from CLL patients relapsing on venetoclax, thinking that this is the best approach to identify mutation and clonal evolution and understand the therapeutic resistance. Then we move to another approach that used systemic genome scale screens. And finally, we characterize match resistant and sensitive lines. So first, we investigated 6 CLL patients with relapse refractory disease relapsing on venetoclax after a median of 16 months on therapy. We perform matched whole exome sequencing on DNA from leukemia samples collected before and at relapse. Cancer cell fraction value was determined for each somatic alteration by using the absolute algorithm and CCF, were then clustered to delineate distinct subclonal population. When we compared the clonal architecture at before treatment here on the x-axis and at relapse here on the y-axis, we note we note a marked clonal shift, but no BCL2 mutation were detected in this study because with the limit sensitivity of 2% with our whole exome approach. No CLL driver mutation were consistently selected with resistance, but for two patients, as you can see here, patient 1 and patient 2 have a common evolutionary trajectory with TP53 and SF3B1 mutation and leading, and at top of it, loss of 17p, 8p, and amplification AQ was seen, but nothing really consistent and that can underlie the resistance for all the patients. So we move to another approach that is related to genome scale screens. So these technologies have emerged actually as powerful tools to identify genetic drivers of drug resistance. The principle is to induce a tremendous diversity within the cell lines using genetic perturbation technology and to systematically determine which genes provide survival advantage to a given drug. As approach 2, we perform parallel genome scale loss of function and gain of function screen using respectively a genome scale CRISPR screen, sgRNA, and ORF library, which shows to the BCL2-driven OCI-L11 lymphoma cell line that we exposed to venetoclax 100 nanomolar over a 2-week period. So here are the duplicate results showing the top candidate from the CRISPR and ORF screen. As expected, venetoclax promoted deregulation of BCL2 family members, as underlined in red. This showed up in each screen in accordance to their pro- and anti-apoptotic function within the BCL2 family. For example, our CRISPR screen picked up BIM, BAC1, and our ORF screen, MCL1, BCL2, BCLXL. And of great interest, our screens also reveal novel hits. Indeed, as you can see here, among them were well-known lymphoid transcription regulators, underlined here in green. And interesting, most of them are also known to be recurrently mutated in lymphoid cancers. The unexpected candidate hits from the ORF screen, here underlined in pink, includes component of a separate but related AMP-dependent pathways such as PRKAR2B, a subunit of the protein kinase A, and PRKAA2, a catalognic subunit of AMPK, a key regulator of cellular energy metabolism. So to better understand the consequences of this deregulation, we generated as approach 3 a venetoclax-resistant OCI-L11 through chronic in vitro exposure to the drug. And this allowed for the assessment of venetoclax resistance signatures and the availability of a tool for functional studies and in vitro drug testing. So, as shown here, here is represented the expression profiling and the differential expression at the transcript level on the x-axis and at the protein level on the y-axis. And here are the differential expression genes. So between resistant and sensitive lines. So the first finding was that among the overexpressed genes, MCL1 emerged as the only significantly and coordinately deregulated transcription protein that also overlapped with the gene hits from the genome scale screens. So we confirm its overexpression in by using Western blotting. And in addition, we show that if you inhibit MCL1, there is a synergy with BCL2 inhibition, and also the MCL1 inhibitor can restore sensitivity to the BCL2 inhibition. So when we look at a patient, we also confirm in several cases that we can show an increased expression of MCL1 by using immunohistochemistry at relapse compared to before therapy by venetoclax. Interestingly, in some patients, the baseline MCL1 expression was already high, and this predicts in this very small series response duration to venetoclax. So this first result shows that MCL1, increased MCL1 expression is responsible for venetoclax venetoclax resistance and can be efficiently targeted. So another set of findings were related to cellular energy metabolism. In this same differential expression profile, we have seen some metabolic regulators that are dysregulated, and also some could be linked to the genome scale screens. First, because the lymphoid transcription factors have shown to act as metabolic gatekeepers, preventing the transformation in more aggressive cancer. And in addition, AMPK is a key regulator of cellular energy metabolism and oxidative phosphorylation. So that's why we asked whether or not energy metabolism and oxidative phosphorylation changes are venetoclax resistance mechanisms. So here is represented the electrotransport chain at the inner membrane of the mitochondrial membrane, and there is several complexes, and a way to interrogate this mechanism is to use inhibitors such as antimycin and oligomycin. And we also use this seahorse assay, where the oxygen consumption rate were measured over time and after inhibition by several of the compounds I just mentioned. And this data actually revealed that the OCL-L11 resistant cells have increased basal and maximal mitochondrial respiration than the sensitive ones. So this is for one cell line, and we have also replicated these results for other cell lines. So again, we check whether or not inhibition of oxidative phosphorylation synergizes with venetoclax, and we use oligomycin and other compounds to reveal that yes, when you inhibit oxidative phosphorylation, you synergize with venetoclax in several cell line models, and you also restore sensitivity to venetoclax in the resistant cell line, and that was true in vitro and in vivo by using a xenograft model. Finally, to go further on the mechanisms that are involved in this energy metabolism network, we characterize several cell lines that were repressed for several of our best candidate hits. And we found the greatest similarities between the resistance signature we have generated by comparing the resistant versus sensitive OCL-L11 cells to the signature we get by regarding the transcriptional expression profile between ID3 and control sgRNA lines, and also some other target. But here, the best similarity was found with ID3. That's why we focused on ID3. And what we found is that if you look at what was the top hit of the regulated transcript when we repressed ID3, you actually get PRK-AR2B. And again, if you overexpress some of this, the same transcript, you get a repression of ID3, and this is paralog ID2. So these were the first step to highlight a network implicating ID2, ID3, and PRK protein kinase A signaling as a network regulating the energy metabolism in venetoclax resistant cells. Again, we checked in patients, and we also found that in tumor samples collecting after venetoclax when at the time of relapse and at baseline before venetoclax, that you have an increase of the expression of AMPK, and also of the AMPK signaling is next is the downstream effector being ACC, and so you have we can see here that there is an increase also of phospho-ACC in some cases. And in 3 patients, we were able to get sufficient RNA and sufficient quality because these were FFPE samples. And we have checked that there is also an ID3 repression in some cases. So at the end of the day, here is the model we propose to explain the resistance to the BCL2 inhibitor venetoclax. First, at the outer membrane, we have an overexpression of MCN1 that is able to sequester beam, so that the venetoclax is not active anymore. There is no permeabilization of the outer membrane. And that the inner membrane, there is also a mechanism that involves increased oxidative phosphorylation levels that are under the control of AMP and protein kinase A, ID2, and ID3 transcription factor. So what we are doing now, we are trying to investigate larger cohorts because you have seen that we have only few patients, and that is also true in other work worldwide. So we are collecting samples across France, and we are trying to assess the potential clinical relevance of our findings. And we're also interested in another characteristics of this disease, chronic lymphocytic leukemia, that is this disease, one way to escape therapy is to transform into a more aggressive lymphoma that is called the Richter syndrome, and we are characterizing this disease right now. And we are still trying to find collaborators and funds, even more important, for going further on mechanistic aspects for these projects. So thank you very much for your attention. I would like to thank, of course, the team in Clermont-Ferrand, the shelter team, as you can see on the left, and also the very important collaborators for this work at Dana-Farber Cancer Institute. That has been directed, this work has been directed by Catherine Wu, and you can see her Chinese last name, and also at Broad Institute, Gali Getz and Vamsi Moussa. And I have also further collaborators here in Clermont-Ferrand on the next projects, Luca Valser at the GRED, and other collaborators at Metabolism Platform here in Clermont-Ferrand. Thank you very much.
Thank you, Romain. Thank you very much for this talk and this wonderful talk. It was very complete, and actually the signaling pathway is highly regulated. We can see that. And actually there is Hichem. Okay, so Hichem Mertani from Lyon is asking a question. So yeah. BCL2, I don't know if you are able to see the question. So BCL2 being frequently overexpressed in various cancers, have you already tested venetoclax effect in other adenocarcinoma?
So on my side, being an hematologist, I'm just focused on hem-onc, but for sure, this drug will be evaluated, is actually on evaluation in many cancers. And I've just talked about CLL, but is that it has been recently approved for AML, so acute myeloid leukemia, together with another compound. So I don't know actually for adenocarcinoma, but for sure there will be one of the trial testing this, because it's such a rational therapeutic target for many cancers.
Okay, I have a question actually, Romain.
Yeah.
Actually, I try to recapitulate how the different target, you have new target like ID3, for example, which could be a new target of the venetoclax. And actually, I was wondering, because BCL2 is more or less more dedicated to mitochondria signaling pathway, I was wondering how you connect ID3 and this mitochondrial signaling pathway.
Yes, so so there is several way there is nothing very close and very well demonstrated, but what we know from our data, at least, is that when you repress IT3, you have an overexpression of the PRKIR2B, that is a regulator of the protein kinase A, that is related to the energy metabolism at the end, and the mitochondria is at the end of this signaling pathway. So that would be, this is what we so far think, but there is maybe many other possibilities. So
yeah, but actually, I have just if you are affecting the PK signaling pathway, if you upregulate the regulatory subunit of PKA, you are supposed to decrease the PK activity.
Totally, yeah. So yeah, there is directional difficulties with this. So what we are testing now is whether or not all the balance between AMPK and PKA is actually in play in lymphoid cancer cell, and it's not necessarily as clear as we think from the metabolism and very simple. So another aspect is that this protein kinase A also has other aspects than the kinase. The kinase signaling we know, and especially this subunit can also phosphorylate other targets than those from the signaling pathway, but we still don't know which one. But I agree with you. It's not, the story is not that simple, for sure.
No, I know how signaling can be complex, actually. Okay, if there is another question from HM. How to prevent possible alteration of mitochondrial activity in normal cells by venetoclax treatment?
Okay, so so it's this is related to the compartment question for each target therapy, but in this the cancer cell we are studying, there is an increased expression of BCL2, very high, and much higher than in normal cells. And in addition, these cells are super dependent on BCL2, and normal cells are not as dependent on BCL2. And by using even very low concentration of this compound, you can restore the apoptosis in a target way on the cancer cells and not on the normal cells. That being said, we use every day this compound in clinics, and what we see is that there is no that many adverse events. One is important is that there is toxicity on other blood cells, but not on other organs or function. So it's a well-tolerated drug, actually better tolerated than other compounds we used to use in this leukemia, such as chemotherapy. So this is what I can say right now. Another thing that I just want to clarify, I did not have time to talk about this, is whether or not venetoclax affects directly the metabolism in the cells. And the answer is yes. Actually, we have tested this, and venetoclax induced a rapid decrease of respiration. And again, here we don't know the complete mechanism, but we have recent data showing that it is related to the mitochondrial permeabilization. So you still need to have the mitochondrial permeabilization to induce these consequences of venetoclax on oxidative phosphorylation. But so maybe we will have consequences in cells on metabolism, on normal cells metabolism, but that is still not known.
Okay. Okay, so thank you very much. And thank you, Hichem, for participating to this
And thank you for this interesting question. These were all interesting questions.
It's not so easy to participate through question by webinar. But anyway, thank you, Romain, again.
Thank you. Thank you to